name is Mirna. I'm doing my PhD in bioengineering, and one of the topics that I'm really interested in is antibiotic resistance. So today I'm going to be talking about a research paper that was published by a group at MIT where they used AI to discover new antibiotics. So just to give you an idea of why this is important, we have a huge problem in the medical field right now that is driven by antibiotic-resistant pathogens. Those are basically bacteria or microorganisms that infect you, but they're not easily killed by available antibiotics. And today, we have 2.8 million US infections per year. And unfortunately, globally, 700,000 people die every year because of antibiotic-resistant pathogens. And if we don't address this very soon, this is going to be turning into a pandemic. By 2050, if we don't come up with a new solution to this problem, we expect 10 million deaths globally, which is crazy. Unfortunately, no new antibiotics have been discovered in the past 25 years. And just to give you an idea about how we find new antibiotics in general, usually we would collect samples from the environment. Those samples will contain microorganisms like bacteria, fungus, yeast, different types of tiny organisms that have what we call secondary metabolites. These are like chemicals that they secrete, okay? And the way we have been finding antibiotics so far is by screening whether these secondary metabolites that these microorganisms are producing can kill harmful pathogens. People have been deploying this method for a long time, and we kind of hit a plateau because we started discovering the same molecules over and over and over again. No new molecules have been discovered, and that's because there's a lot of redundancy in biology. So there's a lot of common pathways and common metabolites between different microorganisms, and that's why it's really hard to find anything new. So this group at MIT, they are led by Professor Jim Collins. They decided to try to use a combination of AI and experiments in order to try to discover new antibiotics. And this paper here, this is a news article and it's based on a published research paper if you are curious to read it, but it's basically the first time a new antibiotic has been discovered using AI. So here's their approach. The first thing is they decided to train a deep neural network model. Then once they had the trained model, they decided to start applying it to new chemical libraries, so apply it to new chemicals and molecules that were never looked at as potential antibiotics. And once they had that model applied to new chemical libraries, they decided to start testing out whatever the model is giving them as suggestions for antibiotics in the lab. So they have to make sure that actually works. And there are also a few different criteria that you have to think about when you choose which candidates to test in the lab, because experiments are expensive and there are other limitations that I'm going to be talking about in a minute. But here's the model overview. The training data set basically consisted of around 2,300 molecules. Okay, these are all molecules that have a known antibacterial activity. So we know how effective they are at killing pathogens. And you can think of that as the label, the efficiency or the effectiveness of the molecule at killing E. coli specifically. That's our label. And then the training data set is basically just the molecules. They contained 300 FDA approved antibiotics. So these are already exist, right? As well as 800 natural products from different organisms like plants, animals, and microbes. But we know their activity, their antibacterial activity. So great, so now they have their model. And one thing to note about this, the reason why they took a slightly different approach from normal is basically, usually when people have chemicals, they try to extract features from the chemicals, okay? So they try to look at functional groups or mechanism of action of the chemical, things like that, and they will input that as a feature into the model. What this group did is they decided that they're not going to extract any features from the molecules. They're just going to give the model the chemicals. They're not going to try to analyze the chemicals, describe the chemicals to the model or anything. And this is because they want to avoid any assumptions about which chemicals usually work. Because if you talk to a chemist, they would basically look at the chemical structure and they would say, okay, this chemical structure has these functional groups and I expect it to behave in this specific way. So there are some assumptions that are made, right? But because they want to discover something new, they decide that they're going to go without any assumptions about chemical groups or mechanism of action or anything. And that's really powerful. And we'll see that in a minute, that the molecules that they discovered are actually very different from what the conventional molecule or antibiotic looks like. So once they had that trained model, right, they decided to first apply it to drugs that are currently under investigation for a lot of different human diseases. And the reason why this is important is because when a chemical is being, or like a drug is being developed, that takes a lot of money. And unfortunately, many times after companies invest a ton of money and time into developing a drug, it turns out that that drug doesn't work out, because when they test it on an animal, usually it doesn't work out in a human. But the main thing that I want you to keep in mind is that when people try to develop new drugs and they bring them to clinical trials, even though they don't work, the one thing they know about the drug is that it's safe. Okay, and that's a huge thing. Safety is a huge thing. So if we have like a ton of molecules that we know are safe for humans to consume, and even though they are not working for the intended disease, I think it's a good idea to look at them and see what else can we use them for, right? And this is what they tried to do. They were trying to see if any of the molecules that are being in clinical trials can be used as antibiotics. And from this approach here, they came up with 100 different candidates that they tested in the lab on mice. And it turned out that actually one drug that is currently being investigated as a diabetes treatment is actually a very good antibiotic. And that antibiotic is called halicin. So halicin is actually the first antibiotic to be discovered using AI. And one of the cool things they observed is that when they took it to the lab and did their own experiments, they didn't see any resistance to the drug. So usually if you expose the same pathogen to the same drug over and over and over again, to the same antibiotic over and over and over again, typically the behavior that you see is that the organism will start to mutate and will become resistant to the antibiotic. This is how we have antibiotic resistance. But from the testing that they did in the lab, they didn't see any resistance. Of course, I totally believe that they have to do more, but like with the experiments that they have, no resistance observed. And one thing that it is worth noting is that the mechanism of action of this drug is actually very different from the existing antibiotics. And this again comes from the fact that they decided to make no assumptions about mechanism of action or function of the drugs. So not to go into details, but basically the way this drug works, it basically disrupts the flow of protons across cell membrane and just messes up the whole growth of the cells and kills the cells. This is not a typical antibiotic mechanism of action. Awesome. So they discovered this drug and they decided, okay, they didn't want to stop there. They decided to expand the prediction space of the model. So instead of giving it just 6,000 molecules under investigation or clinical trials, they actually decided to look at the whole database that has like 100 million molecules. They fed it into the trained model, and they came up with 23 different molecules that they decided to test in the lab. And out of the 23 compounds that they tested against different bacterial pathogens, 8 were actually very effective at killing one of the tested pathogens, at least one of the tested pathogens that they looked at in the lab. And one thing to note here, similar to halicin, these molecules actually have chemically, they have very low similarity to known antibiotics. Again, eliminating the bias initially really helped look at these new antibiotics that weren't originally thought to be effective. Yeah, so again, rather than searching for specific structure or molecular class, they just looked at molecules and their particular activity, no assumptions made. They were able to find molecules with new mechanisms of action, molecules with new structures that were unexpected. And this is how they are harnessing AI to give us new antibiotics. Now, some things to think about when using AI specifically in the sciences is how do you choose the few molecules that you are gonna bring into the lab? I'm pretty sure when they inputted the 100 million different molecules into the trained model, they got a few thousands, right? Few thousand good predictions for compounds, but they only went with 23 compounds. Obviously, like time limitations and things like that are one factor, but there are other factors that they had to look at, and they had to come up with criteria on how to pick different compounds to test in the lab. So obviously, they needed chemicals with high prediction score. They also looked at structurally unique compounds. So instead of looking at things that are similar to current antibiotics, they intentionally looked at the ones that the model suggested that were not similar to current antibiotics. And another thing that's very important is that they picked compounds that are unlikely to display toxicity, because you don't want to kill the animal while trying to treat it for an infection, right? We also have to think about different limitations in terms of getting like these predicted compounds that the model tells you, okay, you should try these compounds, right? So a few things to think about, like when they chose these compounds is, first of all, are we able to synthesize these molecules? So imagine you find a drug that's amazing, but you go and you find out that you actually cannot make it like on a large scale, maybe due to technical issues or chemical reasons, or even cost. This is not very useful in the long run, because we always want to bring whatever we discover in the lab into real life, right? And if again, this has to do with toxicity. If the compound is unstable in aqueous solution, which means it's unstable in water, that disqualifies the compound, because we are made mostly of water, right? And if we consume something that's not stable in water, that's toxic to us. So the main takeaway here is that AI can actually be very powerful, especially when we remove bias in giving us new drugs, right? But we always have to come back to the lab and do all the experimental work. And we have to look into whether whatever the AI predicts is actually something that is viable in real life. Can it be done in real life? Can it be done on the large scale? So especially in sciences, I think in all fields, but especially in sciences, because that's my field, I would say machine learning should be used as a tool, and there's a lot to do with it. But it should be used as a tool in combination with experimental results, right? So that's it for this research paper. I just want to highlight a couple more AI-driven advances in science in case that's something of interest. So Google AlphaFold, which is basically a model that can predict a protein's 3D structure just from the amino acid sequence or the DNA sequence. This is very powerful because we couldn't do this before. This was very hard. And the reason why this is very important is because a protein's 3D structure actually dictates its function. And if we want to understand everything that's happening in the body or in the environment, we have to understand the function of the proteins that exist in that environment. So this was very powerful and something that really is revolutionizing the field of bioengineering and protein engineering. The other thing I want to highlight is that there are a lot of companies right now that are using AI to fuel bioengineering efforts. One of them that I think is pretty cool is called Arzeda, and they're basically trying to design and make proteins that don't exist in nature, like new proteins with new functions. Everything is new, all with the using AI.